Good afternoon. My name is Rui Costa, and I am a professor of the Polytechnic of Coimbra, a researcher of the Research Center of Natural Resources and Environmental Society, also in Coimbra, and Secretary General of the Executive Food Association, based in Austria. And first, I'd like to thank the organization of this summer school for inviting me here to this summer school. It is a pleasure to be here, although at a distance. Fortunately, I already had the pleasure to be in Almaty, in Casnaro, and so it's almost like I'm there. So I will present about the mass transfer modeling and macrostructural changes during soaking of pulses. I don't know if this is a topic that is interesting for you. I hope it will be. So I will start speaking about pulses, what it is, and later on I will speak about the typical pulse processing. Then I will focus on soaking, the main operation that I will deal here. After that, I'll speak about the structure of pulses and how on the water pathways during the hydration. And finally, I'll finalize with the hydration kinetics. So first, the first part of my presentation is about what are pulses. From this picture, you already see what I'm referring to. Chickpeas, beans, lentils, and green peas, for example, are pulses. Okay, maybe this is a term that you're not used to. But let's speak first about a bigger family where the pulses belong to. I'll speak about the grains first. So there are several families of grains. And the grains are defined as being plant seeds that are produced for purposes other than that of plant reproduction. They are part of a healthy diet due to nutritional composition, and especially in legumes. There are these four groups of grains. A bigger group that are the legumes, the Fabaceae family. Then another smaller family that are the cereals, the Poaceae. Then the Amarantaceae and the Cucurbitaceae family. I will now give a little bit more detail. So first, let's go the family of cereals in terms of their content, in terms of nutritional content. They are relevant because they contain around 8 to 12% of protein and 55 to 70% of starch. So you all know that we get mainly calories from starch from these cereals. The second family is high in protein, which is not so common. Also, pumpkin seeds and this third family are high in protein and lipids. They are also not so common. Finally, the family to which I will speak, it's the legumes family. They are high in protein, around 18 to 25%, and in starch also 55 to 65%. Notice that these are intervals, and in some papers, some scientific reports will find slightly different intervals for these contents. But anyway, if you think if you keep this idea that legumes have around 20% of protein and 60% of starch, is something already good to identify the difference to the other grains. And so just one note here is that there are more than 20,000 species of legumes, but only a few dozens are eaten by mankind. This is something curious. So pulses. So the pulses now are part of this last family that I spoke, the legume family. They grow in pods. So like here, you see, this is a pod. They grow in pods and come in a variety of shapes, sizes, and colors. They have annual crops yielding from 1 to 12 grains or seeds. And now what we call pulses are the legumes with a low fat content. So for example, we exclude here peanuts and soya. And so the crops are harvested solely for dry grain. And so it excludes crops harvested green for food. We're not talking about those. Crops used for oil extraction, like we said, soybean or peanuts, and also leguminous crops like seeds of clover, alfalfa. So soybeans and peanuts are sometimes referred as leguminous oil seeds. And so they are not pulses. So pulses are all the legumes that do not have a high fat content. According to the Food and Agriculture Organization, since 1994, so this could probably be updated now, there are 11 types of pulses: dry beans, dry broad beans, dry peas, dry chickpeas, cowpeas, pigeon peas, lentils, bambara beans, vetches, lupins, and others that are not specified. But here we can include, for example, grass pea, which I will present. So pulses are relevant for several reasons, not only for nutritional purposes, but also because they enable to fix nitrogen from the air to the soil. And this is a highly important characteristic that supports a good crop rotation. So it's good to cultivate weeds alternated with a crop or a legume crop that will enrich the soil in nitrogen, and so on. Also, there's another characteristic of legumes is that they need very little water. For example, compared with soybeans, they need 20% of the water, and with peanuts even less, almost 1/10. And if you compare with meat or animal products, they require much less water. So let's watch a video to have some sound and some music here. Oops, sorry.
Pulses are an awesome superfood that you've probably never heard of, at least not by that name. Most likely you've eaten them before. Pulses are the ancient name for dried peas, lentils, lupins, chickpeas, and dozens of dried bean varieties that have been a worldwide staple for centuries. Your grandmother's split pea soup? Yep, pulses. Hummus? Pulses again. Chickpeas or garbanzo beans are the main ingredient. Iranian dal adas is a simple red lentil soup and it's delicious. The point is, pulses are the newest ancient superfood because the benefits of this amazing food are just starting to gain attention. But in countries like India, Pakistan, Egypt and the United Kingdom, pulses have been a food staple for aeons. An affordable food loaded with protein, fibre, vitamins and minerals, pulses are good for the plate and good for the planet. Farmers grow them around the world because they're one of the most sustainable crops in the history of food. That's right. Pulses require little water and fertilisation to grow, and they're a good neighbour, leaving nitrogen in the soil and enriching it with the nutrients needed to help other crops grow better. Healthy, nutritious, affordable and sustainable, pure and simple. Pulses. For a healthy body, for a healthy world.
Okay, so let's continue. Sorry. So pulses around the world, you can see from this map where pulses are cultivated, almost every part of the world. But the biggest producers are India, Canada, Myanmar, China, Nigeria, Brazil, Australia, USA, Russia, and Tanzania. And the biggest exporters are Argentina, France, Ethiopia, and Turkey. How about the pulses in history? They started, at least it's written in a Greek poem from Homer, before 800 years before Christ. So there's a long history record that they have been used for thousands of years. And nowadays they are gaining importance, and in 2010 they duplicated their production compared to the beginning of the century. And to 30 years before, and it is expected that this will double soon. What about the production of pulses in Kazakhstan? According to this website, knoema.com, they are around 356,000 bones. You see, of course, they are much smaller than compared to India, China, but already comparable to Pakistan, Bangladesh, Nepal, etc., and your neighbors, Kyrgyzstan, Uzbekistan, and Tajikistan, which in fact they are higher than the production of these countries. Compared to other crops that you grow in Kazakhstan, of course, it's much lower than cereals, fruits, and vegetables. And I suppose that you don't eat so much these foods, the pulses, and so it's natural also that you don't produce so much. But I think nowadays the trend is to increase the consumption of pulses, and so it's probably a good idea that Kazakhstan can increase the production of these cultures. So in terms of nutrition, what is the value of pulses? I already said they are high in protein and carbohydrates, especially in starch. They are not high in fats. Sorry, there's a mistake here in this slide. They are high in dietary fibers, B group vitamins, and minerals. The composition can vary, obviously, according to cultivar, location of growth, climate, environmental factors, and soil type. This is common to every culture. So starch is the main constituent of pulses. It complements well with cereals. So a meal for vegetarians, if you use a cereal and the pulse, it will equilibrate the composition in terms of essential amino acids. And they are very poor sources of fat, also which also is needed in our diet, but especially omega-3. They are very poor on fats, and so very poor sources also of fat-soluble vitamins, and although they are rich sources on water-soluble vitamins. They are also excellent sources of minerals, calcium, phosphorus, potassium, sodium, etc. So what kind of processing exists, or it is usually done on pulses? That's the second part. There are many studies on this, not all of them conclusive. I just present here a table of one, where you can see that boiling, oven baking, microwaving, steam boiling, blanching, freezing, pressure cooking, etc. They are used on pulses. And some I highlight that there is a high retention of the nutrients during this processing. And so here there's not a clear, of course, then there are some advantages of the processing to decrease anti-nutritional factors that this process may contain. In general, they have to be cooked in order to be eatable. So, but at home we can use this kind of process, but I will refer now to what it is done, other kind of processing more usually used at the industry, that's roasting, extraction, malting, fermentation, germination. So in this case, they all use pulses that harvested as dried, which in one case it's good because if we have it dried, we can have a long shelf life. But when they have to be processed, they have to be hydrated, usually as being soaked. And that hydration, it usually is the first step of something. It can be the first step, for example, of fermentation. So that's how the soya sauce is done. So I'm giving here an example of a fermentation of soya, but others can be found. Although soya, as I told you, is not exactly, is a legume, so but it's something similar to pulses. What about the germination? So germination is the transformation of a seed into a plant. So here are the several steps that occur in germination. Of course, usually we don't do this at home, but some people do it. Why they do it? For example, if you have eaten sprouts from soya, that's how they are done. We have the seeds of the sprouts, and then we can germinate it. So we have to humidify it, hydrate it, and then germination occurs. What happens in germination? So of course it's used mainly for development of new plants, but for example can be used for the sprout production that then can be served as a food immediately. And what happens here is two changes that are relevant in terms of nutrition. One is an increase in protein during germination, and then another is the decrease in nutritional compounds. Another example of these processes is the roasted pulses, and you probably know about leblebi. It's a Turkish product or original, but you can find it in many countries, of course, around Turkey, the Balkans countries, for example, um, the Greece, and I don't remember, maybe Kazakhstan and their neighbors. Okay, for all these processes, a first step is needed, is to hydrate. Usually this hydration is done by soaking. As you can see here, soaking, it's just the immersion of the pulse in water mainly. It can be also in another solution, a salt solution, or even a sugar solution, but it's typically in water. So in terms of soaking, we need to improve, to optimize this operation. And that's the topic of my lecture. We studied, but we don't have all the answers for this. But we want to study soaking for what? To know if we should use water or a salt solution, for example. Which volume of solution or water should we use, this ratio to legumes we should use, which ratio we should use, sorry. Then which temperature we should do it, how long, the time, and should we change, for example, soaking solution or not. What we know is that during soaking, there are several things that we need to take into account. First, of course, the uptake of water. That's what we want. Also, we may want to add some other ingredients or not. And second is the loss of solids, including anti-nutrients. This loss of solids may be good if anti-nutrients are being lost to the solution, but maybe other nutrients can also be lost to the solution, which is not good. And as you can see at the naked eye, then there is an increase of volume, and that is clearly visible, related to the uptake of water. Now I will give a few slides about soaking. So as you saw, soaking is the immersion of the pulse in a solution. This solution or water, it can be simply water, it can be, for example, salt solutions. And in this case, we can have two results, salting. So we supply water plus a little bit of salt, and so the food will become a little salty. But if the percentage of water is very high, we can have osmotic dehydration. This doesn't happen in pulses, but happens if when we do it in codfish, for example, in meat, in products with a high water content. The same with sugar solutions. If we use sugar solutions, we can add a little bit of sweet. But if we use sugar solutions, for example, in fruit, we will extract water. And so that's what we call osmotic dehydration. So during soaking in general, we have water, definitely, then we can have salt and or sugar and or others, and then or some acid, for example, or alkaline molecules to change the pH if it is favorable for our process. So soaking is not only, when you speak about soaking, is not only about immersion in water, it can have some variations. And in pulses, there are applications of soaking in water and soaking in salt solutions. For example, for leblebi, it's produced in several ways. One of them is to soak in salt solutions, even very high concentration. So here I just made a plot to observe that if we hydrate in grains, whatever the sodium chloride content, we will have an hydration as a result. But if you hydrate fish, meat, or non-dried vegetables, we have an hydration, but for a large content of sodium chloride in the solution, but we can also have dehydration. So this is something for you to pay attention if you're going to use salt solutions to soak a product. If you soak products that already have a high content in water, if you soak it with salt solutions of at a very high salt content, you will probably cause dehydration. Okay. So what happens during the hydration in soaking? Let's assume that is basically made with water. Water enters and solids leave. And so there is extraction of solids and there is uptake of water. Of course, the water content increases, then it can germinate if we leave it for a long time, and if the cells are not dead. It will soften the texture and it will cause some reactions. After hydration, then we have other things that are changed. After hydration, the heat transfer is faster, so we can cook it faster. It facilitates reactions that need a very high water activity, and if we cook it, we can cook it in a shorter time. What happens on extraction? On extraction, we can see extraction of nutrients like vitamins, starch, and protein, and others. But there's some extraction that we would like to see. For example, of toxic alkaloids from lupin, or extraction of phytic acid, tannins, phenols, and trypsin inhibitors that are present in many of pulses, and they are called anti-nutritional components. But although this is a little bit controversial, some people say that okay, this is the cause of a decrease, the cause of less value of nutrition for pulses, and others say that this is not, in fact. So also, some pulses sometimes they result in flatulence, and they say that they can be by these factors or part of these factors. And others say that no, if you are used to it, you have the way to process it, the microbiome to process it, so there will be no problem. Anyway, most of the researchers indicate that this extraction of anti-nutritional components is important, and so soaking can be used for that also. What kind of reactions can we see? After hydration, activation of cell wall enzymes that results in better polysaccharide solubility and shorter cooking time. A more homogeneous gelatinization of the starch and the naturation of proteins during cooking. The heat transfer is enhanced, and also the inactivation of these anti-nutritional compounds. Also, it's something that can happen. Notice that they can be lost they leave the pulse and go to the solution, but they can also be inactivated when heated. And now, these are 3 points, 3 general reactions that happen after hydration. Now, I will speak about the hydration in detail. How the water enters the pulse. So for this, we need to look at the structure of the pulse and how it enters. Let's look at the raw pulse. This is a green pea. This picture was obtained by tomography. And you can see here the green pea has a seed coat, this is skin, and it has two cotyledons, okay, one here, another here. And you see that there is a big space between them. Okay, so there are several spaces here. Here in the top, between the skin and the cotyledons, between the cotyledons, and also here between the skin and the cotyledons. Now let's look at the legume structure. Here is the image of a bean extracted from this paper from Miano and Augusto. So I already spoke about the seed coat and the cotyledons. The seed coat represents around 10% of the total weight, while the cotyledons represent the remaining 90%. So you see here, there is a seed coat, and also there is here some holes, the hilum, the half, the micropyle. Here you can see it in another way, how it is cut. So the seed coat is more brown, and the yellow is the cotyledon. And this, the hilum, it's here expanded. I'll show you more detail, and you see that is a hole. And if it is a hole, the water will go here inside. See from here. Here is a picture, more detail of the skin. And here is of the cotyledon. I will show a better picture after this. So here you can see on the bean the three structures where the water can enter: the hilum, the micropyle, and the other half. Here again. And now if you look at this in more detail, the cotyledons, this is a very nice picture where you can see also the skin, and here the cotyledons. You can see the cell walls here. Yeah, they see these lines. You can see what is in blue, dark blue, the starch granules, and in green, the fibers and the proteins. Okay. You can see they all fill the rest of the cells. So the cotyledons are these reservoirs of starch, fiber, and protein. What will happen? How the water will go in? So here we still have some pictures about. You can see these at the magnifying glass spaces between the skin and the cotyledon. This is for the chickpea. This is for the grass pea. See here a picture on the left of grass pea. The same thing, some voids. And still inside of the cotyledons there are air spaces because these products are dried. So we can see here, I will show this in more detail. You see that these darker parts are holes, empty spaces. You can see here bigger spaces without any material, and close to the center there's less air spaces. So water will enter to these empty spaces here and also to these ones. And so usually we measure these empty spaces. We call it porosity. if the porosity is 0, it means there's no empty space. If it is 100%, it would mean that everything is empty. So if we have bigger holes here, these white parts, we will have a high porosity product. If you have less holes, it will have a low porosity. We could see that the porosity is around 7% in chickpea and 14% in grass pea. So this will affect how the hydration occurs. What happens when we hydrate? If you hydrate at a high temperature, then all these products, all this material will grab water, will expand, and then if it is boiled at high temperature, the starch will gelatinize. And then if you dry it again, it will not come back to the initial shape. It will stay like this. Look here. This is the raw green pea, and this is the dried after cooking green pea. You see that the starch was gelatinized, was expanded, and then when it dried again, it didn't retract to the original shape. So there is less space in between than there is when it was raw. Okay, what happens in terms of hydration kinetics? We have first a water uptake. We have 3 stages: water uptake until the maximum possible, without germination. It's stage 1. Then we have some time where the germination is starting. So this part after the stage 2 and stage 3, usually it's not needed. It's not needed for fermentation. It's not needed for lab production, etc. So most of the products will stop here. But if you want germination, then we need to go over this stage 2 and enter the stage 3 where there is germination. And so here, just wanted to say that this stage happens in dead cells or in live cells. If it happens in dead cells, we'll never reach this stage 3 germination. If it happens in live cells, then we can have stage 2 and stage 3. Now, how the water enters. So first it will enter the helium, as we saw, and then it will go through the center space. It will go to the spaces between the skin and the cotyledons. This is the first. The second step. When the seed coat is completely hydrated, then the water continues to enter to the helium and follows these steps, this direction, and also from the outside to the inside. Because the skin is usually has some fat around it, and it's a little bit impermeable to water. So that's why first this step, entering the helium, occurs faster, and then when all the skin is wet, it will become permeable to water, and only then we'll have this third phase. So this is represented here and here. The same thing. So always enter the helium, go under the skin, and after that it enters everywhere from every side. Now, what kind of curves of uptake of moisture along the time happens? Of course, there are 2 different curves. One curve that is called the downward concave shape. It's this one. It's like, like this. Only the water goes up quickly and then continues to go up, but not so quickly until it stops. And there's the sigmoidal shape, where the water is entered very slowly, and then at a certain point it starts to enter more faster, and then it starts to enter again slower. So this is the way the water always enters, but it doesn't. Sometimes this part is very fast, and we don't have this first phase. Okay? So, but if this first part is slow, we will have this first period of the sigmoidal curve. So the water enters slowly, and then at a certain point starts to accelerate the rate at uptake. If the initial moisture is higher, these curves are affected. For example, we can have a sigmoidal curve for a very low initial water content of the pulse, but if the pulse, the water content of the pulse increases before hydration, you may see disappear this first lag phase and only have the downward concave shape. One thing this is important. It is important to speak about hydration is the glass transition and isotherms. I don't know if you are all familiar with it. I will not spend much time about it, but just to say that every product usually can be in two states: a glassy state or a rubbery state. Imagine potato chips. They are usually in a glassy state because they just break. When you buy it and you take it from the pack, you can just break it. You cannot mold it. So it's in a glassy state. If they gain a little bit of water, if they become humid, you can mold it. So that's on a rubbery state. Let's see here the example of pasta. This is pasta, noodles. They can only break, they cannot be deformed. If you touch them, if you try to change their shape, you will break it. Okay, so it's in a glassy state. But if it gains a little bit of water, it goes from the glassy state to the rubbery state. That's what happens when we cook. And so when we cook the pasta, it becomes that we can change the shape. It becomes the rubbery state. So that's what happens also with the pulses when they gain water. They come from the glassy state to the rubbery state. Usually that happens for those that are aware of isotherms. It happens. Isotherms has three kinds of shapes. This is the most typical. It happens in this value of moisture content and water activity. Okay. Here it's when it is changed from the glassy state to the rubbery state. Okay. So there are authors that made these studies and saw certain kind of beans have follow the sigmoidal shape of uptake moisture content along the time. Others, they follow the downward concave shape, and so you can find this in the literature, and it can be interesting then to choose a mathematical model to predict the time, the evolution of water content along the time. And some already studied, let's say, families of hydration, which kind of pulses, for example, here, follow this downward concave shape hydration, which kind of pulses follow the sigmoidal hydration. So there are lists about it. Just one thing about the live and dead seeds. Dead seeds usually absorb more water. So dead seeds are those that cannot germinate. since the living seeds have a turgor pressure that counteracts hydration. But this, the effect of the grain metabolism in this stage is still needs investigation. Now, I will enter my last part of the presentation is about the hydration kinetics. I have here a video just for fun. So I'm talking about hydration kinetics of pulses and not of cats, but I found this interesting to present. So in terms of hydration kinetics, what do you want to know? You want to know, want to predict how long it will need to soak a chickpea to achieve a certain water content that is suitable for my process, for roasting, for example, or for boiling, whatever. So we need to know the kind of behavior of hydration kinetics. We need to know the mass transfer and fluid flow mechanisms in the process, and which mathematical model it is better to apply. So the mathematical model will help me to predict the moisture content as a function of time, to help me to study the process characteristics, the hydration rate, the equilibrium moisture content, and lag phase time. These mathematical models, they are usually two types: empirical, so we can use any mathematical equation, or they are based on physical laws that usually consider the process as diffusional or capillary flow. But I don't know also if you are familiar with it, so I will not extend a lot on this. Just to show you the type of equations that we apply if we have a downward concave shaped curve. You see here the Fick's second law. It's this one. It's this one. Yes, this. It can have different aspects. A look, let's say, according to the shape and the conditions, external conditions of mass transfer. But and this is not a line. So Peleg model is here, and the first order kinetics equation is here. So if you look at this equation and that one, they are quite similar, just they don't have this equation in the first order kinetics doesn't have the sum and these terms, but this part is very similar because here in the F it also contains the time. So these are the models used for this DCS hydration, and for the sigmoidal hydration, we have usually the capsule model. It has this look, and the Ebers-August model. There are other models, but these are the most used ones. So the hydration of mass transfer, how it occurs. First, it occurs because there is a difference of water activity that is the driving force. So the water is transported from a substance with a high water content, the soaking water, to a substance with a low water content. It usually is the pulse. There's a complex structure inside the pulse, as you saw. They are not isotropic. It means they are not equal from the surface to the center. Different tissues, cells, different sizes, zones with different permeability, etc. And the water does not only enter by diffusion, as we saw. If it enters the ileum first, enters first by the capillary flow, and then later on by diffusion. So it's a mixture of mechanisms. Okay, we can apply those models to the soaking. Here are some examples, the case of soaking chickpea. First, before showing the result of the model, just to say that the soaking, what happens during soaking. Imagine that circle in blue in the top here, it's like a chickpea. It's curious that when water enters the chickpea, there is first a decrease in volume. You see, on the top, I will show it again, the simulation. There is a decrease in volume, and then there is a decrease, okay, until the end, okay? So you can see here, the mass always increases, but the volume, you see here from these points, decreases initially and then increases. And of course, if the density, that is the mass divided by the volume, it will increase first and then decrease. We can do this at several temperatures. This is a plot at 25 Celsius degrees, but we can study this at 50, 75, and 100 degrees, and we can see always the same behavior, and decrease of volume, and then an increase. And we can see that the higher the temperature, the higher the mass, and the higher the volume we obtain in the end. So we will obtain chickpeas with a higher mass and higher volume if we hydrate this at 75 degrees than if we hydrate it at 50 degrees, for example, or if we hydrate at 25, especially. Okay. If we apply these models with the diffusion models to predict how the water content leaves, we can see this here. You see, this is like the water content. These are the experimental points, and the curve is the predicted curve. You see that there is a high variation of experimental points, but the curve predicts pretty well how the water content develops. And the same for the solids loss. There is less solids loss, but it still can be predicted well. Something that is curious, and we should talk, I should draw your attention, is if we plot the water content versus the fraction of initial solids in the beginning, so it starts soaking here, there is an increase of water, so it goes in this direction, less and low solids, but at a certain point, there is a high solids loss and little increase of water content. So we should stop the process around here. So it's good to make the plot like this, water content versus fraction of initial solids, to see the point where we should stop during soaking, where we are losing a lot of solids and not gaining much water. So this is relevant, in my opinion. So since I already took a long time, I will go through this plot and just call your attention that the soaking of chickpea in salt solutions was also studied, and which is relevant, for example, for lab production. And we can see that if we increase, if you salt it at 1% of salt, we will gain more water than if you salt it at 5% or 20%, which makes sense because if we have high salt content, we don't have so much water in the solution. But this difference is quite dramatic in terms of volume. In terms of volume, we have a high value of volume increase at 1%, and this is even higher than at soaking in water. So soaking in 1% of salt helps the results in the chickpea with a higher volume than soaking in water, which is good for process gains, for example. We can have a heavier product in the end and sell it, sell more mass than we have if we have a lighter product. In the case of grass pea, sorry, we have similar, we apply the similar models. The difference is that the data in grass pea vary much more than in chickpea. The replicates present a very high variation, and the errors are enormous. In this case, if we do again the fraction, the water of the fraction by the fraction of solids, we have a similar plot and a similar conclusion. We should look to this plot to decide when to stop soaking. One thing that we saw in recipe is that the smaller the seed, the smaller the seed, it means the higher the area per volume, the smaller the water content at equilibrium. Well, but these are certainly details that are not so interesting for you as the initial ones, especially if you're not familiar with this. And so I will stop here and hope you enjoyed, and I'm open to collaborate with you or send you some material if you need. Just contact me on this email. And I wish you continue to enjoy the summer school and the training in Casnaru. So it was a pleasure again to be with you, and hope to see you in presence in another year. Bye.